Hello and welcome back for part 2 of neurons and synapses. So last time we focused on neurons and how the action potential moves along the neuron and down the axon to the axon terminal. But what happens after that? Now we're going to be focusing on synapses. So synapses are the junctions where the neurons meet each other, where you've got the axon terminal of the initial neuron. Okay, so let's see. Axon terminal of, for example, this neuron that I was drawing before. Okay, and then the dendrites of the next neuron, or potentially effector cells. So for example, we could be talking about the synapse between the receptor cells and the sensory neuron and the interneuron or the relay neuron. Or we could be talking about the synapse between the relay neuron and the motor neuron, or between the motor neuron and the effector. Those would be the synapses in this circuit right here. I'm going to play a video clip that explains this process of what happens to transmit that signal from one neuron to another across the synapse. So essentially, the general overview is that the signal starts off as being electrical in the axon and then to the axon terminal. Okay, the axon terminal is also known as the presynaptic membrane, so it's the membrane of the neuron before the synapse, before there is this tiny gap between one neuron and the other. So how does it go from being an electrical signal and then bridging that gap and then being an electrical signal again in the next neuron? That's what synaptic transmission is.
The nervous system is composed of billions of communicating cells called neurons. Neurons conduct electrical signals, or action potentials, along the cell's membrane, along the axon to the terminus.
So as you can see here, you've got the axon from one neuron leading up to the axon terminal that's then connecting to the dendrites of another neuron.
The synapse consists of the membrane of the presynaptic cell, a synaptic cleft, and the postsynaptic cell membrane.
Okay, so this is the first neuron that would be receiving the signal, and then it's going to eventually somehow bridge this synaptic cleft, send the message across the synaptic cleft, and begin a new action potential electrical signal in the postsynaptic neuron, which is the next neuron that it's transmitting the signal to. The synaptic cleft is the space between the presynaptic neuron and postsynaptic neuron. This whole image is the synapse.
Since an action potential can only propagate along the neuron's membrane, a chemical known as a neurotransmitter must bridge the gap between the adjacent cells. The presynaptic cell releases the neurotransmitters into the synaptic cleft, where they will move to bind the receptor proteins in the postsynaptic cell membrane. When the action potential arrives at the synapse, it opens voltage-gated calcium channels, resulting in an inward flow of calcium. The calcium binds to the vesicular membrane. This binding activates.
Okay, so take note of what just happened there. The action potential that we talked about before reached the end of the neuron, the axon terminal. As it reached that, what was able to open up? It was the voltage-gated calcium channel. So there's another voltage-gated channel involved here, and that's calcium. So if you ever wondered why you need to eat potassium, sodium, and calcium, this is one of the major reasons why, so that your nervous system can function. So what happens? Why do we need the calcium channel to open? Because of the voltage from the action potential. We need it to let calcium into the neuron so that it can then bind to this thing here called a vesicle, which contains the neurotransmitters. Okay, notice what happens when it does bind to that vesicle.
Activates the vesicles to fuse with the presynaptic membrane. As fusion occurs, neurotransmitter molecules are released into the synaptic cleft.
Okay, so the vesicle, which is just it's a little transporting membrane inside of cells that just is able to contain and store materials and transport them. So in this case, it's storing the neurotransmitters. Because the calcium binded to it, it was then directed to go to the presynaptic membrane, fuse with it, and then release those neurotransmitters where? Into the synaptic cleft.
In the cleft, the neurotransmitter molecules diffuse throughout the interstitial fluid in the cleft. Some of the neurotransmitters will cross the synaptic cleft and bind with receptors in the postsynaptic membrane.
Okay, so it's important to note that once the neurotransmitters are in the synaptic cleft area, they just diffuse from high concentration to low across the synaptic cleft and then bind to receptors on the postsynaptic membrane of the next neuron.
Binding produces an effect in the postsynaptic cell by opening the protein channels and allowing the flow of ions into and out of the cell. If the effect of the neurotransmitter excites the postsynaptic membrane, an excitatory postsynaptic membrane potential, also known as EPSP, is generated.
Okay, so notice that once the neurotransmitter binds to the receptor, that's actually a sodium channel that opens and allows the sodium to enter. So this should start setting off some alarms in your head and maybe remind you of what we just did with action potential. What happens when sodium comes into the cell? What happens to the membrane potential? It should increase. And if it increases enough past the threshold, what happens? Depolarization happens and the action potential is propagated along the axon and fully transmitted. So essentially, this is how the signal bridges that synapse, the synaptic cleft, the gap between the neurons by sending the neurotransmitters out. Neurotransmitters bind with a postsynaptic membrane, receptors, okay, sodium enters, and the action potential starts all over again in the next neuron. Now, the next thing that you need to focus on is what happens to these neurotransmitters after the fact. The IB wants you to know that.
This localized depolarizing signal generally causes long-distance signals, action potentials, in the dendrite of the postsynaptic cell. Neurotransmitters influence the postsynaptic membrane electrical state for only a short time and are removed from the synaptic cleft in one of three ways: diffusion away from the receptors, enzymatically degraded, or recycled by the presynaptic cell.
The ones that you need to focus on for what happens to these neurotransmitters is enzymatic degradation and recycling. In fact, there's a combination of that occurs. There's a combination of that occurs in acetylcholine that the IB wants you to know about. Acetylcholine is a specific neurotransmitter.
Some of the neurotransmitter molecules diffuse out of reach of the neurotransmitter receptors. Some neurotransmitter molecules are removed by enzymatic degradation. Some neurotransmitter molecules are transported back into the presynaptic cell by neurotransmitter transporter proteins, recycled.
Okay.
An example of a specific neurotransmitter that functions in
those transporter proteins that bring the neurotransmitters back into the cell, make sure that you know about those. These are called reuptake pumps. The IB does want you to know about those.
In these ways is serotonin from the receptors and either transport the acetylcholine.
Okay, acetylcholine is really important for the IB as well. They want you to know specifically about acetylcholine, the neurotransmitter acetylcholine. Now, that is a neurotransmitter that is typically involved in muscle contractions and sending the signal to your muscles so that your muscles then can contract. We're also going to talk about how is this related to the death of bees, because the IB also wants you to know about that.
First neurotransmitter discovered also diffuses across the synaptic cleft and temporarily binds with its specific membrane-bound receptor on the postsynaptic cell.
Okay, so similar to what we saw before with the general neurotransmitter example, the neurotransmitter acetylcholine is activated and released in the synaptic cleft due to the calcium that had binded to the vesicle, and then the acetylcholine binds to the receptor on the postsynaptic membrane, and the ions, the sodium ions, go into the postsynaptic membrane and transmit the signal. All of that is the same. But now you have to pay attention to what happens with the degradation and recycling of the acetylcholine.
Affecting the cell's electrical state. Some acetylcholine in the cleft will interact with the enzyme acetylcholinesterase, which will hydrolyze the acetylcholine molecule into acetate and choline.
Okay, so notice what happened there. There's an enzyme that broke down acetylcholine into acetate and choline. Notice what happens to the acetate and what happens to the choline.
Acetate diffuses away, but choline interacts with the presynaptic membrane-bound transporter to be recycled.
Okay, so the reuptake pump actively transports that choline back into the presynaptic neuron so that it can then be recycled. So half of it gets recycled, and then it gets rebuilt into acetylcholine, and you're about to see that.
In the presynaptic neuron into new acetylcholine. Unlike serotonin, which is intact and biologically active following its uptake into the presynaptic cell, new synthesis of acetylcholine is necessary, with the recycled choline as the precursor.
So what is that whole process called? It's called synaptic transmission. Here's a diagram of the overview of what happens there. You should be able to label each of these pieces and annotate them as well. So if we look here, A is going to be, oh, by the way, you can pause it now if you don't want to see the answers yet. Okay, so A is going to be the action potential that has moved down the axon and reached the axon terminal. Okay, and that is what stimulates the calcium channel over here to open. The calcium enters, binds to the vesicle with the neurotransmitters inside of it. That then directs the vesicle to go to the membrane and fuse with the membrane and release the neurotransmitters into the synaptic cleft over here. Once the neurotransmitters are there, they're going to diffuse across, and by the way, this happens through exocytosis with the vesicle. Neurotransmitters diffuse across, bind with the receptors on the postsynaptic membrane. That then directs these sodium channels to open, bringing the sodium in, raising the membrane potential, and starting a new action potential in the postsynaptic neuron. Now, there are a couple other things that we haven't really looked at here, such as there's a calcium pump here. This is an active transport process, so it requires ATP to pump the calcium back out of that presynaptic neuron. Why do we need that to happen? Why do we not want calcium always in that neuron? If your response is that we don't always want neurotransmitters being released into the synaptic cleft, you would be correct. That is because we don't want the calcium to be in the neuron unless there's an action potential, unless the neuron is firing, right, and transmitting a signal. Otherwise, it's going to be transmitting signals when it should not be transmitting signals. We only want the calcium to be entered in through the voltage from the action potential that opens the calcium channel. And then we've got another thing over here. This is the reuptake pump that I was mentioning before. That's the pump that brings the neurotransmitter and recycles it, brings it back into the presynaptic neuron so that it can be reused at a later time. Here is a label of all of the pieces that we just looked at before. And here is an annotated version of that same diagram. Example of a question, a past paper question that you could see about synaptic transmission. So you can pause now before I show the mark scheme. And here is the mark scheme. Here's another past paper question that could be good to also review the different methods of membrane transport that are used throughout this process and just review membrane transport in general. So you can pause here, and I'm going to show the mark scheme. Here's the mark scheme. Okay, so here's a diagram specifically with acetylcholine, because the IB wants you to know about what happens to that specific neurotransmitter. So as a brief overview again, the acetylcholine is activated by the calcium, just like we talked about before, to be released into the synaptic cleft by the vesicle. It binds with the receptor. Okay, the receptor then, the sodium channels then open, allow sodium in, transmit the signal. Then what happens to that acetylcholine afterwards? It gets enzymatically degraded by the acetylcholinesterase into choline and acetate. The acetate just diffuses away, the choline gets brought back into the cell, and then recycled and rebuilt into acetylcholine with the help of acetyl-CoA, okay, which was made and produced from cellular respiration in the mitochondria, if you remember that as well. And now, what in the world does this have to do with bees dying? Okay, you need to know about this last statement here about neonicotinoid pesticides. So, the statement reads, blocking of synaptic transmission at the cholinergic synapses in insects by binding of neonicotinoid pesticides to acetylcholine receptors. What does this mean? It's basically saying that this pesticide, known as neonicotinoid pesticides, which are widely used all over the world, are actually blocking acetylcholine receptors in bees. And this is causing the bees to have seizures and have paralysis and die. Okay, so how is this happening? Well, if we look back at this diagram, basically the neonicotinoid pesticides, which are used on many, many crops, okay, are blocking that receptor right there. They're binding to that receptor and then blocking the acetylcholine from binding to it and doing its job. Okay, this is resulting in overstimulation because the neonicotinoids are not releasing from the receptor and they're not allowing the acetylcholine to bind to it, and then the bees are dying. Okay, this is a giant problem ecologically and economically. Why? Because bees are major pollinators for almost every single crop in the world. If the bees are dying out, which they have been shown to be reducing their numbers and populations by about 30 or more than 30% every year, then this is going to be a major problem for food production and for humans, right? We depend on bees for pollination so that we can have our food products, including even meats, because where are chickens and cows, for example, getting their food from? From those same crops that might end up dying out if we don't have those bees as pollinators.